Hi, everyone, and welcome to today's webinar on challenge studies for acidified foods. We are excited to have you here with us. My name is Heidi, and I'm the Director of Research at Amtech. I will be leading today's webinar. First off, we encourage everyone to participate by asking questions throughout today's presentation. You can submit questions by clicking on Q&A at the bottom of your screen, and we will stay on after the presentation to go through the questions that came through. To start off, for those of you who don't know, Amtech is an accredited laboratory in the San Francisco Bay Area, specializing in food safety testing and special research projects such as shelf life, challenge, validation, and spoilage investigation studies. Amtech also just opened 2 new laboratories in Modesto and Salinas, California to better serve our clients. So if you or anyone you know is looking for superior service in the Central or Salinas Valley, please do not hesitate to reach out. So with that, let's get started. So in the time today, we will discuss a number of items related to acidified foods and challenge studies for this category of foods. So first, we will go over the classifications of acid foods and where acidified foods fall in this category. We will look at the food safety concerns associated with these foods, along with the regulations and requirements for this category of food based on the product parameters and processing. So this will then touch on recommendations for challenge studies and execution. And this will then lead to a summary or discussion of the requirements for study design and ultimately approval of the report by the state and FDA. And lastly, we will summarize the outcomes of the study and the final report that will be used for submission and approval of the product. So when we talk about acidified foods, there are specific parameters that classify foods into this category. So let's look first at the overall umbrella category of acid foods. So acid foods are a food with a natural pH of less than 4.6. A natural pH is the pH of the product prior to processing. So some examples of these foods are going to be yogurt, tomatoes, strawberries, and these foods are generally not regulated. Another category is low acid canned foods. So these are foods that have a finished equilibrated pH of greater than 4.6 and a water activity of greater than 0.85. So the products are packaged in hermetically sealed containers, and this is going to include your shelf-stable foods such as canned vegetables, canned soups, canned stocks. These are all going to fall into this low acid canned food category. And third, we have acidified canned foods. So these are composed mainly of ingredients that have a moderately high natural pH, to which an acid ingredient, so vinegar, lemon juice, they're added to bring that final equilibrated pH to 4.6 or below. So this is essentially low acid foods, that middle category, to which acids are added to have a product with a final equilibrated pH of 4.6 or less. Water activity for this category is still greater than 0.85. So some examples here are going to be canned tomato sauces, canned salsas, relishes, hot sauces that are not fermented. There's a little asterisk about that. Fermented foods are exempt, and we'll talk about that a little further. So both low acid canned foods and acidified canned foods must be produced under the California Department of Public Health Food and Drug Branch under the Cannery Inspection Program. So all products and processes must be evaluated by the University of California Lab for Research in Food Preservation. This is a process authority in California for being adequate to control Clostridium botulinum risk prior to initial production. So I will refer to specific requirements for California in this discussion, but if you're in a different state, you can find the process authority for your state, and there's likely a very similar process to follow along with state regulations and approvals. So for the low acid canned foods, these are regulated in 21 CFR Part 113. So as we talked about, these are foods with a pH of greater than 4.6, a water activity greater than 0.85, and they're considered low acid under this regulation. So the heating process must be determined by a qualified thermal process authority So in California, you must submit a request for official sterilization process. In this form, you will then receive an official process letter from the state, which can be used to file this product and process with the FDA. To fall under the regulation of an acidified food in 21 CFR Part 114, you need to have three things. So first, you need to have a shelf-stable product and a water activity greater than 0.85 that we talked about. So this essentially provides a condition where organisms can grow. Thus, if your water activity is less than 0.85, you would not fall under this regulation. And then the third thing you would need is a pH of 4.6 or below. So there are a few exceptions to this regulation, including things like the naturally acidic foods that I talked about. So canned peaches, you have carbonated beverages, jams, jellies, or preserves. So these are defined by a standard of identity in 21 CFR 150. So these would also be exempt. Some other exemptions, some dressings and condiments, and then microbially fermented foods. So these traditional fermentations, such as sauerkraut, they would be exempt from this acidified food regulation. They're going to have other regulations, but they would be exempt here. So if you are not sure about whether your product is acidified in California, you would submit a request for pH control form, and that would start the process to determine where your product falls, and then ultimately the regulations that would be associated with it. So when we look at food safety, the main factors for these food types relate to first the pH. So a pH below 4.6 is used for Clostridium botulinum control. So improperly processed low acid or acidified canned foods can definitely lead to severe health consequences. So this is due to the growth of Clostridium botulinum. So as it grows in the food, it can produce a potent neurotoxin. So to reduce the public health risk, there are federal and state regulations in place to control production of these types of foods. So we will discuss some of the process and regulation shortly. But in addition to the pH and some of the water activity measures, an acidified food needs to have adequate acidification and be heat treated or held refrigerated. So we have the acid ingredients being added to the product to bring that final pH to 4.6 or below. And this pH will inhibit the germination of Clostridium botulinum spores. So it's essentially that in combination with a mild thermal process. So water bath canning, pasteurization, something like that will then eliminate other organisms of public health concern in these products. So Clostridium botulinum is heat resistant. So the pH control and proper acidification is necessary for control of this organism, while the heat treatment and or temperature control is necessary for killing off other organisms, so your pathogens or spoilage organisms. So keep in mind, refrigerated or frozen foods would not fall under this acidified food category. One thing to note on pH and acidification is that while these foods must be properly acidified to have a pH of less than 4.6, many times they're typically acidified to 4.2 or below. And this is for safety reasons by factoring in that cushion in case of any pH increase in the product and during the shelf life. So while a thermal process step is typically used as a kill step for pathogens, it's not always feasible for products. So for example, maybe the sensory aspects of the product are altered during heating, and thus the product will remain unheated and essentially be cold filled. So this is where there needs to be support by existing data, or a product specific challenge study would need to be conducted. So ultimately, to summarize, in the absence of a thermal process step to destroy spoilage organisms and pathogens, a challenge study is required that determines the hold time to destroy vegetative pathogens. So under the acidified foods category, all products and processes must be evaluated by a process authority for being adequate to control Clostridium botulinum risk. So in California, a cannery inspector would be looking at inspecting each lot or batch after it's produced, but before it's released for sale. So if you have a product that falls into this category of an acidified food, they're in this cannery inspection program. and you would be submitting a request for pH control. So if your product is determined to be an acidified food, or if you're submitting this request for pH control, you will receive an official state process letter, also known as an S letter, and this will provide requirements based on the product category. So the S letter that you get back might indicate, for example, that one, it's an acidified food. So this means that the product was determined to be an acidified food that could support the growth of Clostridium botulinum in the absence of proper process controls. So this product must be manufactured under a valid cannery license. Another indication on your letter could be it's not under pH control due to low water activity. So as we kind of discussed, the lower water activity is going to limit microbial growth. Another one would be not under pH control because it's a fermented food. As we said, these are exempt from this regulation. They would have other requirements for a processed food registration. Not under pH control because it's a refrigerated food. So these products need to be held under 45 Fahrenheit and are thus exempt from this license. Or lastly, you could get an indication back that this is a cold-filled acidified food. So meaning it's an acidified food that was not subjected to this thermal process step to destroy disease-causing organisms and spoilage. So as a result, a challenge study must be conducted to determine the hold time to destroy vegetative pathogens. So if you were to already have a valid challenge study, the documentation could be submitted at this time. If not, additional requirements would be provided, and this is when and where you would reach out to a qualified lab like Amtech to conduct this study. So we talked a lot about the aspects of a product that could factor into it being acidified or not. This decision tree essentially goes through the different questions that need to be answered to determine where your product falls in terms of one, category, and two, regulations. So let's go through this briefly. So if your food is shelf stable, that'd be the first question. We talked about that. If it's not, if it has that temperature control, it's not going to be an acidified food. If it is it one of these food categories that is exempt? Carbonated beverage, jams, jellies, fermented food. Yes, it's not going to be an acidified food. If no, then we're looking at water activity. So does it have a water activity of less than 0.85? If yes, it's not an acidified. If no, then we're on to the pH measurement. Does it have a natural pH before processing above 4.6? Yes, it's not acidified. If not, then we're looking at the main components. So if it's a food composed primarily of acidic ingredients, then it's not going to be an acidified food. It's likely formulated acid, different regulations. If no, if it's mainly low acid with acid food with acid ingredients added, it is an acidified food under the 21 CFR subpart 114. So this just helps to present the different scenarios or questions you would ask related to your particular product. So at this point, you know that a challenge study needs to be conducted for your acidified cold-filled food product. So we will now start to discuss the important and key parameters for the challenge study. So these include the following items related first to the product. So first, we're going to look at product pH. So this is part of the product formulation, and we've talked a lot about product pH and why it is so important for food safety and Clostridium botulinum control. So it is also a very key parameter for these challenge studies. So you're going to want to have documentation and testing as to the range of pH levels your product achieves. So this is going to be your equilibrated pH. And this is because we need to conduct the study at the highest pH your product could achieve for release. So this is typically going to be the lowest amount of acid allowed in your product formulation, and you're going to want to adjust and equilibrate the pH prior to sending to the lab for testing. So we will, of course, test upon arrival to confirm that the pH is still at the target level, but you're going to want to test and make sure it's equilibrated to that pH before we receive the product. Another aspect is the acid level. So there's a lot of information on acids and which forms produce the highest antimicrobial effects and at what pHs. For example, acetic acid, lactic, citric, they all have different effectivenesses against pathogens. So say against Listeria monocytogenes. So this is going to be product specific, and your product should be formulated at the highest pH and with the main acidulant used for this pH control. So the amount of acid needed and the speed at which the pH is equilibrated can depend on many things. And one of them is the buffering capacity of ingredients in the foods. So for example, the buffering capacity of vegetables can depend on the varietal or the maturity. And so this kind of leads into why we're going to want, different replicates in a different number of lots to test. Proteins have a higher buffering capacity, so more acid would be required to lower the pH. So all things to think about, when formulating the food and sending in that final product for the study. In some cases, the product may also have preservatives. So these can be added to help control spoilage organisms, as some of those can grow at pHs below 4.6. Any preservatives would need to be evaluated by the process authority and need to comply with all US laws related to food additives. And then the product processing and manufacturing. So this will include the hold temperature, and we will discuss that key parameter in the next slide. So now we are at the point of designing the specific challenge study parameters. So first, we need to determine the vegetative pathogens that are appropriate for the particular product being tested. This can many times be Listeria monocytogenes, E. coli 0157H7, Salmonella, and Staphylococcus aureus. And we would base the organisms to include off of requirements from the process authority and/or literature in terms of those known to be a target pathogen for the particular product or the pH being tested. So all of these are taken into consideration, and Amtech has an extensive culture collection, and we have a number of strains that we have used in previous acidified food challenge studies. So once the pathogen groups are selected, a number of strains, at least 3 from each group, are selected to be included in each pathogen cocktail. So these should be strains that are appropriate for the food product. So ones that have been isolated from similar foods or have been known to be used in these studies. And the strains are then individually acid adapted through several passes in order to slowly adapt them to the lower pH environment. So this is going to result in strains that are closer in pH to the product and are thus more of a real life scenario of the strains that could contaminate and grow in the product. So this will allow for there not to be an immediate die-off due to exposure of drastic pH differences. So the acid-adapted strains are then combined into a cocktail for inoculation into the product. Another aspect of the study that must be defined is the hold temperature. So this is a very important aspect of the study design. And many times when you ask a client, what is your hold temperature? They say, I hold it at ambient. And so ambient is not a defined temperature and will not be accepted when submitting a final report for review or approval. So this temperature needs to be defined as the minimum temperature, so less than or equal to the lowest temperature your product could be stored or held prior to release. So this minimum temperature is the worst case scenario, similar to the highest pH the product could achieve being the worst case scenario for that indicator. So you would want to monitor the temperature in the warehouse or the facility that your product is being held in order to generate the temperature data to support the temperature chosen for the study. So for, say, an ambient held product, a lot of times 50 degrees Fahrenheit is used as that lowest temperature. More than one product batch should be challenged. A lot of times 3 replicates is recommended. If there is expected or anticipated variation in ingredients, you're going to want to use different ingredient lots to prepare the batches for testing. So the ingredient variability can be due to location of production or the seasonal variability. And so you want to ensure that you're including all of that in the study so that we're testing all the different variables that could factor into the product and its ability to kill organisms. So as the expected variation increases, so too should the number of replicates. But like I said, many times 3 replicates is what is used. Uninoculated controls should be included. So these should be included and tested alongside the inoculated product. So this is to monitor the natural microbial population of the samples. So we're looking at aerobic plate count, yeast and mold, lactic acid bacteria, depending on the product, and also monitoring pH in the uninoculated and inoculated samples throughout the study. And the goal of the study is to demonstrate a 5-log reduction in the inoculated organisms of the product. So, and we want to also determine the time it takes to achieve this reduction. So at least 4 test points should be conducted, and with the 5-log reduction demonstrated for at least 2 consecutive test points. So some of the key considerations to kind of summarize what we went through on the challenge study design. So we're looking at a cocktail of organisms. So we want the organisms to be acid adapted strains and isolated from acidified foods or from similar products to the one being tested. Inoculation of the cocktail should be less than 1% of the product volume. And this is to ensure that the parameters critical to the formulation, so pH level, acid levels, are not changed by the addition of the inoculum. So additionally, we want to ensure a high enough inoculum to be able to confirm this 5-log reduction. And this needs to keep in mind the limit of detection. So for example, you're going to want to inoculate at 7 to 9 log CFU per mil or gram in order to be able to confirm this 5-log reduction. And lastly, typically each pathogen cocktail is inoculated into separate samples. They're not co-inoculated unless this has been tested prior to the study in a pilot trial. It's typically done individually. If you want to test quickly after inoculation as that baseline test point, as there can be some immediate die-off, and you're wanting to make sure that you're catching it so that you have everything ready and are able to, perform that test point soon after inoculation. We need to ensure that media is appropriate. So we're wanting to use non-selective media that should be help to allow for a recovery of injured cells. Selective media can sometimes overestimate the die-off of pathogens. So we're either going to use an overlay method or non-selective media to help allow for the recovery of these injured cells. The worst case scenario conditions. So I've talked about this a number of times, but one more time to kind of highlight the importance, because this is extremely important for approval of the final report and the product, both initially and continued approval. So these worst case scenarios include the lowest fill temperature or lowest fill temperature hold time and highest pH. So these are both important parameters as the pH of the product becomes the maximum pH of the process. And the temperature is the lower limit for the hold time. So if the product during manufacture goes outside of these ranges, either pH or temp, the product will be retained by the state. We are going to include the separate lots. So we're going to do this to test ingredient and batch variability. So that will be tested through 3 is typically recommended. And then the requirement for the 5-log reduction to be met with the minimum number of test points being 4 so that you can kind of monitor that kill curve of the organisms. So the outcomes of the study. So you want to have this demonstrated 5-log reduction if that is required for the study and the product formulation to be accepted. So if this is not achieved, or if the hold time is longer than would work with your process, there would need to be reformulation, and ultimately another study would need to be started with that reformulated product. The final summary report, we would generate that. It would include all of the relevant information that would be needed for regulatory approval. So you're going to have product information, all the materials and methods, raw data, and results and conclusion would all be encompassed in that final summary report. But one other thing I did want to note here is that any changes in product formulation would need to be reviewed. So if you make any changes to the product, then the process authority would need to review them to determine if they would still be covered by the current study that you have, or if another study would need to be conducted based on the reformulation. So we went over a lot of information related to the food category and the challenge study design and the overall process. To summarize the steps, if you were to work with Amtech to complete a challenge study like this would include, first, we'd have an initial meeting to discuss project and goals, as well as the product and processing information. And based on this, we would then design the study. So this is going to be based on the product parameters and the S letter requirements. So your hold temperature, pH, the organisms to include in the study. And then once the design and proposal is complete, we would present this to the client for approval. This design can also be provided to the state if required in order to get their approval prior to starting the study. So we've done this previously as we want to ensure that the study is designed to meet all of the requirements. If revisions are needed, these would be incorporated in order to get to that final design. Once a final design is in place, we're ready to start acid adapting cultures, receiving samples, and initiating the study. So during the study, we are in contact with results, updates, information on the 5-log reduction attainment so that you're aware of how the study is progressing. And we make note of any changes that we are observing throughout the study so that those can be included in the final summary report. At the conclusion of the study, we are then preparing the final report that can be used for submission to the state and to FDA for approval of product release. So with that, I want to thank everyone for joining this webinar today, and I hope you found it valuable. I would encourage you to join the next webinar on October 11th. That's going to discuss common mycotoxins in food. We will now end the recording, and at this point, we should have some time for a few questions. If you have questions that are related to your specific product, you can definitely still submit those. And if we don't get to them, or if we need to discuss in more detail, we can reach out following the webinar to discuss those product-specific questions. With that,